Buffers. In the pharmaceutical system, this chapter is a part of Pharmaceutical, Inorganic, and Analytical Chemistry subject of Pharmacy course. Definition of buffer. Buffer is the system which resists in the drastic change in pH of a solution upon addition of acid or base. Buffer is mixture of a weak acid and its conjugate salt. Types of buffers. Buffers may be a solution of a single substance, like salts of weak acid and weak base. For example, buffer system of ammonium acetate. Buffers can also be a solution of mixtures, like acidic buffer. It is composed of weak acid and its salt. For example, buffer made up of acetic acid and its acetate salt. Basic buffer is composed of weak base and its salt. For example, buffer made up of ammonium hydroxide and its chloride salt. There are two types of buffers based on their uses. First is standard buffer system. Second is actual pharmaceutical buffer. Standard buffer system is used for analytical purpose. It is readily available with desired pH ranges. Actual pharmaceutical buffers are used to maintain pH limits in pharmaceutical formulations. pH limits sometimes essential for stability of drug or isoosmolarity of formulations with body fluids. Buffer action. It is the property of buffer to resist the drastic change in pH, that is, drastic change in hydrogen ion concentration. Definition of buffer action: the resistance of a solution to change in pH upon addition of small amount of acid or alkali is termed as buffer action. Let us take an example as, if you add 1 milliliter 0.1 normal HCl in 100 milliliter water, pH will drastically change from 7 to 3 pH. But if this 100 milliliter water is buffered, say for example 0.01 molar buffer solution with 7 pH, when you add 1 milliliter of HCl, buffer will resist the drastic pH change, and pH level is maintained nearly same. Mechanism of buffer action. That means we have to understand how this buffer resists to drastic change in pH of solution. Buffer is made up of acid. It may be weak acid or conjugated acid of weak base, and base. It may be weak base or conjugated base of weak acid. Because buffer have both acid and base components, thus buffer have ability to accept proton when acid is added in buffer system, and to accept hydroxide ion, or to donate proton when base is added in buffer system. Let's take example of acetic acid sodium acetate buffer system for more clear explanation of mechanism. This buffer system contains acetic acid as weak acid and its acetate salt. If small quantity of acid is added, then sodium acetate reacts with acid and form acetic acid. Acetic acid is weak acid, thus its dissociation. That means reverse reaction rate is low and cannot release proton easily. Hence, acetate ion accepts added proton from the system and maintains the pH of solution. If small quantity of base is added, then acetic acid reacts with base and forms sodium acetate. Hence, acetic acid accepts added hydroxide from the system and maintains the pH of solution. The actual process is acetic acid releases its proton to neutralize added hydroxide from the system and maintains the pH of solution. Let's see example of basic buffer system. Ammonium hydroxide, ammonium chloride buffer system. Ammonium hydroxide accepts added proton from the system and maintains the pH of solution, while ammonium chloride accepts added hydroxide from the system and maintains the pH of solution. Buffer capacity is defined as the number of gram equivalents of strong acid or strong alkali necessary to produce a change of 1 pH unit in 1 liter of the solution. Buffer capacity is also called as buffer efficiency, buffer index, and buffer value. The larger the buffer capacity, the better is the buffer. The more concentration of buffer composition is directly proportional to buffer capacity. Maximum buffer action obtained when the concentration of acid and base are equal. Buffer equation. Buffer equation is equation that gives relationship between concentration of buffer components and pH of buffer. The main buffer equation is the Henderson-Hasselbalch equation, which calculates the pH of a buffer solution. Factors affecting on selection of suitable buffers. Available with desired pH range. Available with adequate buffer capacity. It should in a high degree of purity. It should possess a pH which is minimally influenced by their concentration, temperature, and ionic composition or salt effect of the medium. It should with desired chemical stability. It should not absorb light in a visible or UV region. It does not alter the solubility of other chemicals of solution. Should not precipitate in redox reaction. It should not react with other chemicals of solution. Toxicity of buffers is also concurred selection. For example, borate buffer has systematic toxicity, thus cannot use in parenteral and oral formulations. Only used in topical applications. Buffer must be safe and should not interfere in the pharmacological actions of the drug. Volatile species like ammonia or carbonate should be avoided, since their loose may alter the pH of system. Alkaline buffers should be protected against the absorption of CO2 from air, which produce drop in pH. Role of buffers in pharmacy. Buffer is used in pharmacy in various purposes. First role of buffer is to maintain pH limits in drug preparations. During the storage of preparations, pH may change due to leaching of alkali from certain glass containers, gases like CO2 and ammonia dissolution from air to preparations. This causes poor stability. Thus, buffers are used to improve the chemical stability and solubility of the drug. Inorganic salts like ferric, phosphate, borates are soluble in acidic media, but precipitate in alkaline media, so acidic buffers are used in formulations. Ascorbic acid and penicillin are unstable in alkaline pH. Second role of buffer in pharmacy is to maintain color of preparations. Many natural dyes present in drug formulation have been found to be pH dependent. For example, red color of cherry and raspberry syrups are stable at acidic pH, which become pale yellow to nearly colorless at alkaline pH. Next role of buffer in pharmacy is to obtain a specific pH for analytical purpose. During drug assay or other testing, pH of a drug solution is maintained by appropriate buffer system. Mainly phosphate buffers are used in HPLC analysis of compounds. Standard buffer solution used to calibrate pH meter. Buffer also has important role in patient comfort. Injectable and internal or external use preparations become irritating if their pH is different greatly from their site of application. So pH adjustment is necessity to avoid tissue damage or irritation during drug use. The normal useful range of pH to prevent eye damage is 6.5 to 8.5. Tolerated pH range is 3 to 11. So injectable formulations are prepared within this range and pH maintained in such range. Examples of various pharmaceutical buffers widely used in pharmacy are listed here. Commercially, buffers are available in various forms as powder, tablets, and solution form. Important study questions from this topic. Thank you.